Hey guys, I'm going to share my screen and we're going to do a presentation for lab number 11. Okay, I'm going to try this again because I've already done it twice on PowerPoint and it hasn't appropriately recorded. So hopefully this will work this time. Welcome to lab number 11, which is methods to detect pathogens in food with a focus on DNA sequencing and bioinformatics. So this lab is meant to be partially in the lab and partially data analysis. And of course, we're not going to be in the lab because of our situation. So what I'm going to try to do in this presentation is explain how the lab works. And actually, what you're going to do is some data analysis towards the end of the work associated with some of the sequences that we would produce. So DNA sequencing, just to let you know, DNA sequencing is as it sounds. It's the sequencing of DNA. Bioinformatics is the science or the analytical methods by which we analyze those sequences. So there's three major objectives to this lab. The first is to understand the way sequencing and molecular methods are actually used to determine phylogeny. Phylogeny is the type of organism, whether that's the genus, the species, in other words, where it fits categorically in the phylogenetic relationship of microorganisms in general. In this case, you're going to get sequence associated with an unknown bacteria, so will have no idea what that bacteria is, and also to understand the workflow that's necessary in order to sequence the DNA of microbes. Um, if we were in the lab, you'd have the opportunity to use a DNA extraction kit, which allows us to isolate the DNA from the culture. You would do PCR to amplify a particular gene. In this case, it's the 16S ribosomal RNA gene, and you'd perform a PCR cleanup. Unfortunately, you won't have the opportunity to do any of those because those are lab-based studies, okay, or lab-based work. You then would get the sequence. We would send the sequence out to a sequencing facility and analyze that using online tools to check its quality, to determine what that sequence actually is, and to determine what the organism is that corresponds to that sequence. And that is the part of this exercise that you will have an opportunity to do. So, what is sequencing? Well, very simply, it's the process of determining the order of nucleic acids within a DNA segment. You should already know from our previous laboratory exercises, particularly the one on PCR, that that the sequence of DNA, certain sequences of DNA, are very specific to certain microorganisms or even to certain strains of microorganisms. And if you know these unique sequences, then you can use that sequence to identify the microorganism of interest. And that's exactly what we're going to be doing in this DNA sequencing exercise. There's two general types of sequencing. The first has been around for a very long time. It's called Sanger sequencing. It's very basic, highly versatile, pretty inexpensive, and pretty quick. And that would be the type of sequencing that would be done in the sequencing facility here at NC State. The second is something called next generation sequencing, which is much more complex, but of course extremely powerful. And there's lots of different applications for next gen sequencing. I think the two most important, probably, or the ones that we hear about a lot nowadays, is looking for the entirety of the DNA sequences within a complex sample. Think about a biofilm or the microbiome of the gut, or the nasal passages, for example, which allows us to determine what the microbial composition is of those particular sample types. There's also a relatively new method called RNA-seq, which allows you to determine the sequence of a population of RNA that is in a sample. RNA-seq is great for looking at gene expression in particular, and is a great research tool. And there are many others as well. So, sequencing has applications certainly in more from a food science standpoint, it has applications in certainly beyond food safety. But we're going to talk really specifically about food safety in this lab. So, one of the ways we use sequencing is for routine identification of pathogens. And as you know, I mean, PCR is kind of sort of a type of sequencing. At least it's a sequence identification method. Many times, if you have a PCR product, you might sequence that product to determine that it is indeed the sequence that you think it is. We do that a lot for non culturable pathogens. Um, Increasingly we're taking we're purifying cultures and sequencing the entire genome of that culture. That allows us to identify unique strains that may fall into a genus or a species category but may differ from other strains. And there's lots of other ways we're using this in food safety. A great example, actually, is in troubleshooting persistent microbial issues in a product or a plant. And I think here you might think about listeria, for example. If you have an L mono colonization problem, let's say in your plant, and you know that it's a single strain and it keeps, it keeps popping up all the time. Frequently, these persistent strains will last for years in a plant environment. Then sequencing is very helpful as you begin to work towards eliminating that strain from your environment to determine number one if you've eliminated listeria in general, but number two if you've eliminated the strain that you're most concerned about, which is that, or the strain that you are concerned about, which is that persistent strain. We certainly use sequencing very widely to monitor all types of foodborne pathogens, whether that's in the environment, in animals, or in humans, or in foods. And we use it in outbreak investigation a lot. And it's helpful for many aspects of outbreak investigation, but the two probably main ones are if you find a cluster of patients, even if they're separated spatially or temporally, but they have this very, very same sequence associated with the infectious agent, then usually we know or we suspect that they've been exposed to a common source. That there's a common source of something that's made us sick. Now, it may not be food, it could be another common source. But the bottom line is that that's kind of a clue that further investigation needs to be done, and epidemiologists will launch those types of investigations. Another way that it's been really helpful is in tying a particular food product to a particular illness in a particular group of people. So usually that's kind of the smoking gun. If you can prove that um, you can find that organism in that product that's epidemiologically linked to a group of people who got sick. That's pretty compelling evidence that that was the cause of the illness, and we'll actually talk in much greater detail about that in a couple of weeks when we talk about epidemiology. So just a couple of examples. The target that you use for DNA sequence analysis really depends on the pathogen. So for the pathogenic E. coli, 0157H7 is an example. We frequently designate serotypes or serovars, and that's based on the somatic, flagellar, and capsular antigens. So those would be the genes associated with those would be the target for sequencing designed to type those types of pathogens. For Salmonella, Salmonella enterica is the species that causes most foodborne disease. There are about 2,500 um, subspecies under the Salmonella enterica category, and we're very interested in knowing what that subspecies is that caused illness, whether it's typhimurium or enteritidis. And so we would target our sequencing to categorize those organisms at the subspecies level. And there are also a variety of other methods that are DNA based that are used that either use sequencing or are linked in some way to sequencing, used to classify and track pathogens in many, many different types of applications. And this is a great example, actually, of one of the first times that we really use sequencing in an outbreak investigation. You may or may not remember, Bluebell ice cream was associated with a large listeria outbreak probably about 5 years ago. This was an example where we used whole genome sequencing of both listeria isolates from patients as well as those that were obtained from the ice cream to make that clear link between disease and contamination. The Genome Tracker program, which is a program that was instituted by FDA and has grown pretty massively over the last 5 years, was instrumental in facilitating that. Another example of just kind of shows the resolution of whole genome sequencing or genome sequencing in general is has to do with identification of erythromycin-resistant Campylobacter that popped up in some turkey flocks. It popped up in a species called Campylobacter jejuni, which is a significant cause of human disease. But it's not a resistance that's normally found in jejuni. It's instead normally found in Campylobacter coli, which is a lot less pathogenic to humans. And when they went in and did the whole genome sequencing, what they found is that it was a single nucleotide change between coli and jejuni that was responsible for for the development of this erythromycin resistance. So this is an example of the kind of the degree of resolution that you can get from sequencing for answering questions about microbes, the emergence of certain features of microbes, et cetera, and so on. So the general steps, and again, you're not going to be in the lab, so I'm going to talk about the general steps you're going to use, but you're not going to actually have the opportunity to do them this year, unfortunately. So we start with a pure culture of the organism of interest, and you guys are going to receive an unknown sample. So it's a pure culture of a bacteria that we know what it is, but you would not necessarily know. And that would have been something that would be prepared by the TAs. You then would go through kind of a 3-step process. The first would be to extract out the DNA from the culture. Okay. The second would be then performing PCR on the gene of interest. And the third would be cleaning up then that PCR product so that you've removed some of the reagents in the PCR that might interfere with sequencing. And those 3 steps would have actually been done by you in the laboratory. And then you would send out for sequencing. If we were running, we would send it out to a central facility on NC State's campus. So let's talk a little bit about these individual steps so that you at least understand what you would be doing if you were doing this in a hands-on way. So the first is producing a pure culture, and you guys have worked with pure cultures several times in this lab. Basically, the idea is that you would take whatever your initial culture is, you'd streak it out on a plate, pick an isolated colony, and then put that in a new, clean, and sterile tube and grow that up. And that would form the basis of your sequencing. So the importance of doing this streaking for isolation, I think, is it's critical for you to understand. This is actually not just done for sequencing. This is done for many, many, many microorganisms in microbiology in general. And the whole idea is that you want a pure culture. You want to make sure that what you're analyzing comes from a single clonal origin. And what that means is it comes from a single cell. And making the assumption that one colony comes from one cell. By streaking for isolation, you have a pure culture. Um, then you would do DNA extraction, and the purpose of DNA extraction is because we're only interested in sequencing the DNA, and all the other components of the cell can actually interfere with that process. So, what we usually start with is some step that allows us to lyse the bacterial cell, releasing the DNA. And that can be done by mechanical mixing or chemical reagents or a combination of those things. What happens then is the DNA, but everything else, including a lot of proteins and lipids, are also released. And so, using sometimes chemicals, using special column filters that selectively bind the DNA, and then we elute the DNA off of those filters, we're able to remove lipids, proteins, and any of these other molecules that might interfere with the sequencing reactions. So what's left is relatively pure DNA. And the way we measure the purity of that DNA is with something called the 260-280 ratio. This is 260-280 nanometers. It's a UV absorption ratio. And the way it works is that DNA gives you high absorbance at 280, RNA at 260, RNA at 280, and protein gives you this kind of wonky absorbance over a range of wavelengths. And what is considered good quality DNA is if it has an absorbance ratio of somewhere around 1.8, and probably that's give or take about zero point 0.2 on either side. So that's what we're targeting, to try to get DNA that's pure enough so that it won't interfere with the sequencing reaction. Then we take that DNA and we're going to do PCR and amplify the DNA sequence of interest. Now, the DNA sequence that we use in this lab, and that's frequently used for very basic sequencing when you're just interested in identification of the organism on a pretty generic, maybe genus species level, or even just genus level, is amplification of the 16S ribosomal RNA gene. And there's a number of reasons why we choose that gene. One is that it has this unique combination of areas of conservation. So you can actually design primers that will pretty much amplify the ribosomal RNA gene from almost every organism. But within the middle of that amplified region, there are regions of sequence divergence. And it is in sequencing those regions that you're able to determine what the sequence of what's unique about the sequence that allows you to determine that it belongs to a particular microorganism, either at the genus or the genus species level. Another reason why we use the 16S ribosomal RNA gene is that it's present in multiple copy number in the genome of the bacteria. So there are many copies of it, which means that it's going to be easier to amplify by PCR, and you're going to get a lot of it from a PCR reaction. Of course, then we're going to run it out on agarose gel with ethidium bromide, just to make sure that we've got the band that we're looking for. And you can see here on this gel that there are different bands of different sizes. You would, of course, determine the size of the band that you were looking for. A couple of other things that the agarose gel will tell you is the degree of intensity and the size of that band will tell you how efficient your PCR reaction is. So you want a lot of DNA in your PCR reaction. So hopefully you have a lot of it and a strong band. Also, if you see a lot of subbands, either higher or lower, intermixed with the band of interest, it can mean your PCR was either inefficient or you had a lot of junk there that interfered with the PCR. And so it can also tell you whether, how likely it is that that sequencing reaction is going to move forward with success. So there's a cleanup method that we use to remove the primers, DNTPs, and other unused reagents from the PCR reaction. to get that DNA just as clean as we can for sequencing. That's another thing that you would do in the lab, and then it would be sent off to our central facility for sequencing. Again, I had mentioned before we use Sanger sequencing. It basically takes the DNA along with some DNTPs, and it synthesizes a new strand done in a capillary tube, and you'll get a readout that is based on light detection of of the different bases. And so consequently, what ends up happening is you'll get a readout from the sequencing machine that looks something like this. And you'll see on the right hand side are peaks of different colors. And those peaks generally correspond to a particular nucleic acid. And so you'll read those peaks and determine what that nucleic acid sequence is. And then you'll go in and analyze it using something called BLAST. BLAST is a centralized database, very, very large, maintained by the NCBI of DNA sequences from you name it, from all kinds of different sources. In fact, when an investigator identifies a DNA sequence in a study, they are supposed to actually report that into the BLAST system. And you'll put your DNA sequence into BLAST, and you will receive a list of potential sequence matches and the degree to which those your sequence matches those sequences. And really, you're looking for something that's, if you have high quality sequence, something that's 100% matched. In other words, identical to something that is in BLAST. As I said, you're not going to be doing the lab per se, but we have a series of sequences that we've produced at other times, whether that's been for other classes or in my lab during research efforts. And what we're going to give you are the raw sequences, and then we're going to ask you to determine what those are, and then go into BLAST and identify what that organism is, and provide just one quick interesting fact as to about that organism, which you will receive as an unknown. Jeremy has been kind enough to commit to serving as your resource on this. So he will be sending out your sequences. You'll work individually. And on this part of the project, he'll send the sequences out probably sometime on Tuesday afternoon. If you have questions about the analysis, please contact him directly. And when you finish the analysis, please send him the name of your organism and your one interesting fact. You can all do this by email. And he said he's on tap all week to answer any questions. I'd like to have your sequences in by next Monday at the latest. And so I think that's it for this lab, and hopefully this will run smoothly, and I'll talk to you again next week. Bye.